Concept check 3 on chapter 5 is another problem that deals with allelic interaction. This type of interaction is concerned with different alleles that can be present in one locus. So we know that for blood types we can have more than 2 alleles present in the population, but then each individual will carry only 2 alleles, either they are homozygous or they are heterozygous. Now work through the problem and get familiar with this type of situation. Until now, we have looked at gene interactions in the same locus, but we also know that genes can interact when they are not at the same locus. So in this part of the chapter, we will look at genes that are present in different loci. So these genes are not allelic genes, and what type of interactions that they can have. Two important concepts here are that first, gene interactions can produce novel phenotypes. The other concept is that when genes are interacting in different loci and one gene suppresses or interferes with the phenotype of the other gene that is in a totally different locus, we do not call him these allelic interactions because these are not alleles. So we say that the interaction between these two genes is epistatic interactions. The main concept to keep in mind here is that no gene is ever acting alone. It often depends on other genes in order to do its job. So let's first nail down the concept here of how gene interactions at different loci can produce novel phenotypes. Taking the example of the book of the colored peppers, here we see that a red pepper has dominant phenotypes. So those dominant phenotypes will allow for the red color expression. When you cross it with a recessive pepper that has no color, we know that there's no pigment being produced in this pepper. Okay, we cross those two, we know all of the F1 generation is going to be red. We have no problem there. Now, when we cross the F1 generation of heterozygous offspring, what is it that we expect to get here? Here we are crossing two different genes, so we have to separate the alleles first. This is the Punnett square for the first set of alleles, and this is the Punnett square for the second set of alleles. Now let's look here on the side what these genes actually mean. Let's say if we have a certain precursor molecule here, so this is our precursor, that will receive some form of pigment. When we look at the Y pigment of these Y alleles here, we can say that the dominant will produce a pigment that will have a peach color. Now, this same precursor molecule here can take a different pathway. It can be affected by this other gene that when the dominant form is present, it will produce an orange color. So here we have a part on this pathway that will produce a peach color, and here we have another part of this pathway that will produce an orange color. Now imagine that you have a painter's palette that's my representation of a painter's palette. And in this palette, which happens to be the pepper, if you mix your peach color with your orange color, you actually get a red color. So here we are mixing inside this palette this peach and this orange color, and we are getting a red color. So this will tell you that when this pathway here is at its dominant form, we get the peach, and when this here is at its dominant form, we get the orange, and when they both is pressed together, you get red. Now let's put a little summary here that peach plus orange equals red. But now let's look at the recessive alleles. When you have these recessive alleles present, you produce no color for this pathway. And when you have this recessive allele here present on this side of the pathway, you produce no color. So if we imagine here our little painter's palette, you get no color, or as they represent, cream color, when you do not have any of these pigments. So let's write our summary of other colors here. Y equals to cream, and CC equals to cream. That's easy. Now let's figure out how our genotypic distribution will look like out of this cross. Remember that Y phenotype produces peach color, and that the C phenotype produces orange color. Let's figure out here in how many ways out of this cross we can get our red dominant phenotype. In order to get a red dominant phenotype, we need to get the dominant form of the Y being expressed. So that's going to be big Y and something else, which doesn't matter, because if one of these forms is being expressed, the other one can be homozygous dominant or it can be the recessive. Doesn't matter. Now, for our C alleles here, in order to have the red expressed, we need to have a big C. And also on the second place here, it doesn't matter what goes in there. It can be the dominant allele or it can be the recessive allele. So let's figure out here in how many combinations we can get this. We have here 1, 2, 3 out of 4 and 3 out of 4. That will give us 9 out of 16 times that we do this cross. We are going to get both of these dominant alleles being expressed, which is going to give us our red color. Now let's look at what happens when you only have the peach color here being expressed. So in order for this peach color to be expressed, this orange one has to be out of there. So now we know that for this set of alleles here, you must have the recessive form in order to get your peach outcome. Let's see what type of probability will give us this outcome here. 3 out of 4. And for the recessive here, we only have 1 out of 4. So that will give us 3 out of 16 combinations. Now let's look at how many ways we can get only the orange color expressed. So in order to get only the orange color expressed, the peach cannot be there, which means our alleles here for the peach will have to be our recessive alleles. And for orange color to be expressed, we are going to need one dominant, and for the other one doesn't matter. So in this case we have 1 out of 4 and 3 out of 4, which will give us 3 out of 16 for our orange color here. And looking at the other possibility, which is no color, as we have illustrated here, for no color we only have this combination here, which is going to give us 1 out of 4 for the Y's alleles and 1 out of 4 for the C alleles, which will give us 1 out of 16 combinations. So here is how we get our total distribution of 9 to 3 to 3 to 1. Very important to know how to get to this conclusion. So what this means is that our cross here of our original dominant phenotypes give us new peppers in a distribution of 9 red to 3 peach to 3 oranges to 1 cream. And these phenotypes here appeared basically out of nowhere, but we know that they show up because we were using a biosynthetic pathway in which we had a precursor that could be transformed, let's say, either into a peach color or into an orange color, and when they were mixed you would get a red color. And if these two are not expressed, you get a cream color or no color. This example of new phenotypes is clear.